This is a specific group of organisms in their environment. Let's use what we know about morphological analysis to categorize and organize them so that we may better understand how they survive. The first category can be number of eyes. All but one of these organisms have only two eyes. Thus, this antibody will be completely on its own as an outgroup. Here are the beginnings of our phylogenetic tree. The tree is rooted, and the purple box represents the most recent universal organism to all of these creatures. The yellow boxes represent the traits that separate the branches, as well as the theoretical organisms who first possessed these phenotypes and passed them on. We call these internal nodes, which represent the most common recent ancestor between organisms. The yellow lines represent the OTUs. We will come back to them, but they show the degrees of difference between organisms. Lastly, the green boxes are the organisms we see, called external nodes. Let's continue to analyze these creatures and watch the tree grow. The next distinction is in size. Let's start with the larger organisms. They can be divided further into globular beings or more straight and linear creatures. This is the only big and linear organism, called penicillin. Here are the other two organisms with big and globular phenotype. As we can see, they vary in color, with the white one being a white blood cell and purple being Epstein-Barr virus. Here we have cladogenesis, where new organisms have been formed through splits creating clades. One clade includes the white blood cell and the Epstein-Barr virus in the globular clade. More clades will form as the tree continues to grow. Let's go back to the small organisms that can be broken up by their shape, again, linear versus globular. Here we now have convergent evolution, where organisms have evolved similar traits from different common ancestors. This makes sense since these creatures can be all found in the same environment and are competing for niches. Looking at the small linear organisms, we can split them further. We now have the tiny phenotype, which has split off from the linear clade into another clade. We will come back to them in a bit. Here we have the two small and linear but not tiny organisms. Again, we see convergent evolution, with the white organism being a prion and the purple one being a lactobacillus bacterium. Now back to the tiny creatures, which we will also split up by color. White would be anthrax and the green is TB. Last but not least, we have the small and globular organisms. Again, we can divide them easily by color. Blue is a tardigrade and green is a phlox. And here is our finished phylogenetic tree. We can see that the antibody has not yet had any of the other branches. Thus, this is an example of anagenesis, where an organism changes over time rather than through exact branching. We can also see many different clades and internal nodes, and these can help us determine the relatedness of these organisms. And this is where the OTUs are important. The more yellow lines in your boxes apart an organism is, the less genetically similar. For example, the tardigrade and antibody are 10 yellow spaces away, whereas the tardigrade and the flu virus are only 4 spaces away. So clearly the tardigrade is more related to the flu than to the antibody.